Everybody hear me? Hopefully everybody out there in space can hear as well. Well, I wanted to welcome you here on behalf of the AgBioData Steering Committee. We're so excited to see your lovely faces in this room and online. This is our first in-person meeting in many years. So my name is Leonore Reiser, and I'm a member of the steering committee, and it's my pleasure to be here today. I just wanted, before I forget, shout out and acknowledge Annarita Marrano. Pulling all of this together, and the members of the steering committee that have been so instrumental in making this happen. So thank you guys all so much for that. Okay. Oops. Okay, so what are we doing here today? Sorry. So what are our goals? Our goals are to, first off, share the progress and outcomes from the various working groups that have been meeting over the last year and a half or so. So we'll hear some from the working groups that were in year one and have finished up, as well as the current progress from the working groups for year two. And then very important is to use this opportunity to gather feedback and insights from the group as a whole. So in particular, what we're really interested in is that sort of cross-fertilization across the different working groups. So if you were a member of the sustainability working group, you really have, things that you would like to share with the Data Federation training, this is a real opportunity to be able to give feedback to those different groups. So when we go into these breakout groups, I really want to encourage you not so much to stay with your crew. There should always be somebody from your crew, obviously, in those breakout rooms, but to explore, these other spaces with other groups and offer your insight and feedback. So there will be opportunities to do that. We are also taking this as an opportunity to get to know each other better, to expand our personal and professional networks. And we'll take some time in person here to improve our practice in teamwork and working together. Later today, we'll also have an opportunity to do some strategizing and planning. So there'll be a session, what we're calling the future of AgBioData, which is, sort of where do we want to go from here, both in terms of the RCN and then potentially beyond that. Yeah, that's what I meant. So, how will we meet the goals of this current RCN, and then what are our next steps? So that'll be later this afternoon. And I just want to remind you about this NSF-funded RCN has 4 specific aims. The first of which is to make recommendations for standards and implementation plans for making fair agricultural data. And some of the products of that work include the survey data, the recommendations, white papers, and some web resources. So this is sort of where we hope to be by the time this RCN lines up. Second aim is, of course, expanding our network to include a more diverse and representative group of stakeholders. So we're working to increase the number of member resources, our connections to related efforts like fair sharing and RDA, as well as increasing the number of people who are participating. We have a very significant effort that we're working on is this fair education curriculum with a particular focus on genetic and genomic and breeding databases. And we'll be talking about that actually at ASPD later this year about the curriculum modules that are being developed, the website where you'll be able to access that material, videos, and hopefully workshops going forward. And then a fourth and significant aim as well, the RCN, is this developing a roadmap for sustainability for genetic and genomic and breeding databases. And that sustainability working group was launched in the second year. They've already gathered a bunch of survey data, and we'll hear about the models that they're building and the plans for sort of developing this roadmap. So this is sort of where we're headed. So I think at this point, I'm going to probably turn this over to Annarita to talk more specifically about the logistics of the rest of the day. This is, you can use the QR code, I guess, to take a picture. or follow this link to the agenda that's posted on the AgBioData website. I do want to remind people about we have a code of conduct, so please review that and adhere to that for this meeting. And just, I moved something around, so hold on a second. I just don't want to give me the next slide.
Try keeping the X.
Hmm?
Try keeping the X when the recording is on.
Oh, sorry, that's why. Thank you. No. Okay, well, we're not going to get past that. Apparently not technologically enabled. I wanted to talk about, I seriously don't know why this is not going forward.
Escape them.
Yeah.
The full screen.
Yeah. Oh, I think I missed this one. Okay, sorry. So yeah, that's why we're working the slide. I'm a little tired. It's still early in the morning in California. So just because I want to make sure I touched on this about for the people who are participating virtually, the agenda has these morning presentations and virtual breakout rooms, which will all be remote enabled. So we're going to need volunteers to serve as scribes and help moderate the breakout rooms that happen virtually. If you're having problems with anything, you can always go back to the main meeting room, this room here, and there'll be somebody monitoring the chat in case you're having issues connecting. I think we're going to do a poll about identifying what breakout rooms oops, and there it is. I guess we'll do that now. So why don't we just take a minute to indicate, for those of you who are participating remotely, what breakout rooms you would be interested in joining. So you'll see that there's the first choice, one and two choices are for today's breakout rooms. So the first breakout session, you can join the data reuse, standards for genetic variation or sustainability breakout rooms. And then after that, we'll switch, and there's only two breakout rooms virtually. That'll be fair scientific literature and DEI recruiting. So this is your opportunity to indicate which of those you'd like to do. And you can also, there's one session of breakout rooms for tomorrow. So if you can just indicate your choice for that breakout room. I guess I'll leave that for a minute or two.
I think she can also review after the talk.
Yeah, I think after the talks, you'll have an opportunity to go and do that as well. It looks like just people have responded. Do you want to say more about the logistics?
Yeah.
Sorry, I'm going to pass it on to Annarita.
Yeah, I think when we.
Yes, I will just. Hi, thank you everybody for coming. So as Leonore says, so today we will have one guest lecture, Leach, and Monica will introduce her in a minute, and then we will have a presentation from some of the current working groups. They will ask specific questions that then will be discussed during the breakout rooms. And yes, Leonore said it will be the same breakout room in person and virtual with the same question, but only the in-person breakout room and only the data use, which is only online, will report about the breakout room with a 5-minute talk. So if you have volunteered to be those, I'm looking at time, to be the host or the scribe for the breakout room, you should have received an email with a Google link, with a link to a Google Drive, where you can find a synthesis template and the Google Doc where to take the notes. But if you cannot find it, if you go to the website in the agenda, there is also there the link to access the folder, but I can provide support with that also later. And with this, I think I want to invite them to come, and Monica, and while we are, Jen, she's spraying the microphone. Okay, yeah.
And then also, would you like it on your shirt or on? I probably need to use that first.
Good point.
All right, well, I am very pleased today to introduce Dr. Jan Leach as today's keynote speaker for our presentation this morning. Dr. Leach has had a very distinguished career as a plant pathologist. She's currently a university distinguished professor and the research associate dean at the College of Agricultural Sciences at Colorado State University. She has many accolades under her belt. I'll just talk about a few. She has had the Agropolis Foundation Louis Molasses International Scientific Prize for Agriculture and Food. She has had the American Phytopathological Society Award of Distinction, which I understand is the highest award for that field. And she is also a member of the National Academy of Sciences. Dr. Leach's lab studies how plants respond to biotic and combined biotic abiotic stresses, with particular interest in how bacterial pathogens cause disease, how plants defend themselves from these pathogens, and how environmental stresses affect those outcomes. So you may recognize that these research topics involve around the phytobiome, which is a term that she coined herself in 2013, which is essentially the complex interactions of plants, their environment, and the multitude of living organisms in and around that plant. Along with Kelly Edersole and others, she is a founding member of the Phytobiomes Alliance, which was established in 2016. And it was in this capacity that we invited her to speak today. Today, she will talk to us about the Phytobiomes Alliance, share her experience establishing and sustaining a successful network around phytobiomes research, and inspire us as we meet today to do similar work in the AgBioData Consortium. So thank you, Jan.
Great. Okay. So I want to thank you all for the invitation to speak today. This is great, and this is such an important group for the phytobiomes. So I hope we can figure out a way to connect and collaborate in the future. And I also want to say that I'm speaking. If I get this on my belt. 1 second here. There we go. I'm speaking on behalf of Kelly Edersole, who is the executive director of the Phytobiomes Alliance. And I will not do as great a job as Kelly does at this presentation because I'm a scientist more than a founder of a group, but I'll do my best. So with that, I'd like to tell you a little bit of the history. I won't bore you with all the details, but I think relative to what you folks are doing in your RCN, I think this would be a useful background information. So the whole concept of the phytobiomes started back in 2012. Maybe I need back in 2012 when the American Phytopathological Society, is it the battery?
Have extra batteries if we need them.
Okay.
Or we can use them.
Maybe I don't have it.
I wouldn't have I don't know. Maybe it's
So try that. So this started way back in 2012 when the American Phytopathological Society called together a bunch of thought leaders from across disciplines. And those thought leaders were charged with what research do we need to do to meet the needs of and the challenges for agriculture in the future? And how can we collaborate across all of these disciplines? Shall I switch to the other? Yeah, it might be about it. Yeah, that one is fine. Or two. Sorry, folks. Hit the two microphones too close together. Is this okay? Okay.
Maybe there's should be.
Yeah. Is it on now?
Yeah.
And I just didn't have it close enough. I have a tendency not to do that. Okay. So this group of thought leaders came up with a concept of how do we pull scientists across many different disciplines together in order to solve the challenges of the food for the future. And that evolved into the concept that we needed to look at the system of agriculture more broadly. And being plant pathologists, we were focused on the plant agriculture. So this group charged the APS, the American Phytopath Society Public Policy Board, with taking this initiative and carrying it forward. And Kelly Eversole of Eversole Associates was at that time our Washington, D.C. representative and worked with us and the Public Policy Board to develop this concept. We held a number of workshops, so it wasn't just that one group of thought leaders that came together. There were several workshops held over the next couple of years to develop the concept of the phytobiome. And that concept and how we would use that concept to lead this effort was moved forward into 2016 when we finally completed a roadmap for how that progress would be made and how we would tackle it. And the goal of that, or the vision, was to maximize sustainable food, feed, and fiber production through knowledge of the phytobiomes. Now we carried that paper to Washington, we carried it to different groups around the world. And one of the goals of that in the roadmap was to figure out a way to coordinate the efforts across the many disciplines and across societies around the world. And so that was the basis for the foundation of the Phytobiomes Alliance. And I'll talk a little bit about that in a minute. But our thinking was that while most of us focused on our discipline areas individually, so the plant pathologist, I studied how a pathogen interacted with a plant, with a very linear and a very reductionist approach, we need to focus a little broader, and we needed to go beyond that simple approach into the complexity and embrace the complexity of the agricultural system. So agriculture is complex, as you're well aware, and it is nonlinear. There's nothing about agriculture that can be solved in a linear path. It includes multiple interactions and multiple variables. It includes the environment, as well as the plants, as well as the animals that feed on the plants. And it can be adapted and it can be influenced. And that's why we can make, if we understand this complex system, we can influence changes in that complex system. So the phytobiomes approach was at that time a paradigm shift into thinking about these things as a complex system rather than a simple diagnosis, a simple approach. So let me define the phytobiome for you. So in our system, we're taking the phytobiome are the plant biomes. So in this system here, we have the plant as the biome, the place where this interactions occurred. And it is influenced not only by the soil in which it grows, but it's influenced by large numbers of microorganisms, as well as insects, pests, and animals. And it's also influenced by the climate and the weather. So we know that floods, fires, whatever will influence the phytobiome. And most importantly, we can influence the phytobiome by management practices. We can change the outcomes by altering management practices around the phytobiome. There are lots of ways of looking at a phytobiome. You can look at it in a field, a crop field, where pest diseases and weather influence the outcome of that crop, productivity and the nutritional status. You can think of it as a pasture system, where the animals feeding on the plants are part of that components of the phytobiome because they influence the plant or they are influenced by the plant, importantly. There are vegetable gardens and the different management practices that influence the phytobiome in that, forest systems, as well as our containment agriculture, our vertical ag systems, greenhouse systems, and whatever. All of these are different phytobiomes and are influenced in different ways by how you manage those crops. So the vision of the Phytobiomes Alliance is to understand, predict, control, and control emergent phenotypes within specific phytobiomes for the sustainable production of food, feed, and fiber. And we contend that phytobiome science will contribute to solving the grand challenges facing agriculture. So the Alliance itself was established in 2016, and it is a nonprofit pre-competitive research consortium. It is made up of industry, government, and academic scientists. As I said, it's international. At this time, we have 8 countries who are members, but we're working to expand that. The idea is that this is a coordinating group. It is not a funding agency. It is not a research entity in and of itself. It coordinates research across the different disciplines and across the different things that we, different priorities that we have, and I'll talk about those in a minute. And the executive director is Kelly Eversole of Eversole Associates. Now, I know you're working in a research coordination network, and we actually coordinate some research coordinating the networks within the Alliance. And I will tell you that one of the important parts of coming together with the Alliance was to have an external group like Kelly's group help us with the logistics and the understanding of how to set up this Alliance and how to make it work. And I will say that her group has done a tremendous job of bringing support to the table, which I think is my next slide. We have a number of supporting groups. Some of these are founding groups. We also have groups that have come in and they've worked with the Alliance for a few years and then moved on when they felt that they had moved beyond the needs of the Alliance. But it was really important for Kelly as she helped to pull these groups together. And you'll see we have small companies, big companies, universities, as well as international agencies. So again, the goal or the vision of the Alliance is that by 2050, all farmers, and you'll see the application side to the Alliance, all farmers will have the ability to use predictive and prescriptive analytics based on geophysical and biological conditions. So we'll have to understand those conditions to determine the best combinations of crops, management practices, and inputs for a specific field in a given year. So very applied focus. And to get there, there are a number of steps that need to be taken. We need to optimize sustainability and productivity of those crops through many different mechanisms, breeding practices, as well as management practices. Thinking about adaptive, data-driven on-farm practices, those need to be developed, tested, and brought to the table for the growers. One of the other things that we believe is important is to have rapid site-specific diagnostic tools for the producers that they can use out in the field and get rapid answers. And we think those are on the way. The development of prescriptive crop management techniques. How do we get these, how do we tell the farmers what needs to be applied at what time and in what specific space? The development of resilient crops to the different climate impacts, such as the breeding practices or genetic engineering practices, whatever they may be. And then, of course, optimizing soil health as well as thinking about what is a healthy soil and how do we maintain and manage that, even thinking about carbon sequestration in those soils. So I showed you this before, that this is sort of the concept of the phytobiomes, that it's all of these things interacting and talking to one another and influencing one another. But when we set up the Alliance, we needed to focus in a little bit in order to see what was out there. So we explored what research gaps there were. And we decided that we would take initially, because there was so much interest in the microbiomes because of the human and gut microbiome studies that were advancing at that time. At the time when we started this, even the Office of Science and Technology was promoting microbiome research. So because there was also such a great need and such great potential for exploiting the microbiomes and in terms of crop and human health, that we decided to focus on that and how the microbes are influencing plants or are influenced by the plants, because we know that plants select their microbiomes, how climate influences the microbiomes within the phytobiome, as well as arthropods, insects, and animals, and also the management practices and of course the soil. So with that, we set up a number of research priorities that we would help to coordinate across. Of course, microbiome knowledge generation, which was hugely important, or is hugely important. Another thing that I noticed that you folks are very keen on are standards and protocols. And as you know, it's the wild, wild west out there in the microbiome arena. And so we're trying to wrangle standards and protocols so that when we have discoveries, they're meaningful and that they can be taken to application. Huge importance are the regulatory frameworks, because if, for example, someone develops a biologic, a company, they need the ability to use that product out in the field, and regulations across the world, which we're dealing with here in the international arena, are even more of a wild, wild west than the microbiome knowledge and generation. So in order for these things to come to practice, we need to wrangle with the regulatory framework, and we're working closely with groups on that. Of course, data generation and management, I don't need to tell this group how critical it is to have that high quality data generation and management practices up front. And how are we going to do that? And that's where I could see some wonderful interactions with your group. We're building multidisciplinary capacity by the cross-training in these efforts, and not only cross-training across areas of the phytobiome, but also working with businesses and helping them identify what areas they want people trained in order to reflect and to get these practices out to the field. And finally, the precision and digital ag area. That's where the rubber meets the road, and take these practices to the producers who need that. So some specifics, we're promoting projects that link components across the entire phytobiome. We don't really push our efforts for just a microbiome project. We would take a microbiome project on how the microbiome is influenced by climate or soil structure or plant health or plants or whatever, and promote those projects. One of the important things that we're working on specifically with industry is to elevate a sequence-based classification system for microbes. Those of you who deal with taxonomy of microbes know that they change their name every few years, and that is not helpful. And it's not good when you're in a regulatory environment. So there are members of our group who are working on a sequence-based classification system, which will eliminate the need, or eliminate this changing of names. I think the organism I've worked with has changed its name like 6 times in the last 50 years. So, so we need to check, we need that fixed. Developing microbiome standards is huge, and this is something that in the human microbiome area they've been working on for some time. And we are working with that alliance because we need them to understand that a plant and a soil microbiome are quite different than a human gut microbiome, and we need to consider how those standards are going to apply to those organisms. A field is very different than a human gut. Again, I mentioned earlier that we're facilitating regulatory compliance and studying how we might cross international barriers, fix that problem or come up with solutions, suggest solutions. We are, like you, we have an RCN to coordinate microbial collections and networks, both public and private. This is huge because there are a lot of collections out there that when people retire, they're lost, and we need those for identification of organisms. But even more frightening are the number of microbes that we're isolating through microbiome studies. And how are we collecting or maintaining those, and what should we collect and maintain? Should we maintain the living organism or the sequence of that organism? And finally, one of the things that Kelly is most excited about now are the establishment of linkages across the One Health arena. So working especially in the UK with scientists who are looking at how the plant microbiome or phytobiome alters the gut of an animal that feeds on it, the nutritional status of that feed. So the phytobiome is connected because to the gut microbiome of the organism that feeds on it, and how we might alter that, make healthier animals. To do this, we've currently divided into 4 working groups. One is controlled environment ag, looking at greenhouse and vertical ag production systems. One is our regulatory one, which eats up a large chunk of the group. Of course, the microbiome area, and then the livestock and poultry microbiome area. So people ask, why now? Why are we looking at this system of phytobiomes now? And I think it's hugely because there have been in the last 10 or so years some major technological advances in the ability to the genome-enabled technologies, which have allowed us to probe and understand the biological components of the microbiome, the phytobiome. The advances in the precision crop management systems, such as variable rate technology, the ability to apply what you want in a given site at a given time when it's needed. And finally, or not finally, but the computational sciences, of course, is a major part of this group, with machine learning and deep learning and also quantum computing in the future. And the ability to look at these as a system instead of just one component at a time. So we're looking at the networks and how these things fit together so that we can answer these complex problems. And it's important to note that what happened was this was a convergence of the need for solving these critical problems and the opportunities that these technological advances presented. So what do? We organize international conferences where we have a biennial conference, that biannual conference that rotates between Europe and the US. We have, and those are usually populated with diverse speakers that address all areas of the phytobiome, including imaging, regulatory challenges, rhizosphere science, farm sustainability. We have workshops usually held during major international conferences, and we usually run 3 to 4 per year. We have 2 more this year. One, we had one of the plant and animal genome meetings, and another one in the UK recently. But we have 2 in France at the International Congress of Plant Pathology this year. And we run webinars, so 6 to 8 webinars per year on a broad selection of topics. And actually one recent one that might be of interest to this group was one on the science of team science. And I think we had a wonderful woman from Colorado State, actually, who is the world's expert in that area. And I think that's a wonderful, I've worked with other groups who have used them and know that they're quite useful. So finally, I'd like to pose to this group, how can we engage better to expand our impact? I think obviously we are we are meant to be together. There's nothing about AgBioData that does not impact phytobiome. And so I would like to think of this group to think a little bit about how we can help you, or also what you can offer to us. And with that, I'd like to thank you. And if you need the slides, you're welcome to have them. So, and I'm happy to take any questions if there's time.
Have questions, right?
And I think Kelly might even be online.
Yeah.
Great. So
Here are also questions on the chat.
Are you monitoring those?
Yeah.
There were no questions. There is one person that is interested in mining,
oh,
and so she's talking with Kelly.
great. I knew Kelly would be useful.
Yeah. So I have a question about naming the bacterial strains or other microbiomes based on sequence. And you said that in 50 years, 6 times, the name had changed. And so I'm thinking that looking at the duplication time of the microorganism, which is 3 years, 50 years is a long time, right?
Yeah.
And what will be the rate of the natural sequence variation, and will it qualify to be a different strain in that sense?
And so essentially the question is with the evolution of these microbes, which happens every time they turn over, what will the nomenclature look like in those next 50 years? I think what the idea is that if we focus on key components of the sequence, and I'm not the best person to answer this, but if we focus on key components of the sequence that are essential to the microbes, that the change in those sequences is less rapid, and that we wouldn't put them into a new group based on minor changes. They would just, even now, the taxonomy isn't based on just minor changes, it's based on major changes, which rarely occurs. And
do you have a percentage cutoff? Because, you know, like 3,000, 3K, you know, 3 to 6.
I don't know what the cutoff is, and I will send you the paper if you give me your email address. I can send you the paper that was published by the group that's doing this on what it entails. I don't know if Kelly wants to step in and make a comment on that.
I can try. I don't know if you can hear me. Can everyone hear me?
Yes.
Okay. In fact, you really don't rely on phylogeny in the future if we have a sequence-based classification system, because it's literally the relatedness of the ones and zeros on a sequence. That's how it basically gets driven down. And, you know, part of the theory behind it is that you do take into consideration evolution, because you'd see that relatedness to another species. And so what we've found so far, and we're in the process of publishing our results, some negative, some very positive, but we'll be able to show you that in some cases, you can literally, just by a difference of a sequence, like in one allele, you've got a different organism. And in some cases, as what we've been working on is Ralstonia, which is a biosecurity agent, with just that one change in the allele and in the sequence itself, you actually can determine whether something's going to be pathogenic or not. And so that's really kind of the beauty of it. The challenge long-term is combining that information with phenotypic information. And that's really where it's the biggest challenge. And even in our project, which is small, we've dealt with about 143 strains of Ralstonia. We've sequenced all of those, but then we've also have phenotyping studies on top of it. And so with that, it takes a long time to do that. But that's one of the, at least in my view, because I'm involved in the project and in the PI on 2 of those grants, the goal is really to get to a point where we can start using things like deep learning. Can we start making extrapolations and not having to sequence everything? Getting back to what Jan had mentioned, are there ways that we can use a part of the sequence, a part of the genome, and actually see what it comes into as an essential trait and whether it falls then into another area, even though its taxonomy is very difficult, but K-mers just don't cut it. And that's not in the long term. So I hope that's helped and not confused. Did I put my video up? I did. Uh-oh.
I would say that it's hard to get the biologists to accept 1111011101110 over xanthomonas aureus.
Yeah.
That's the hard part.
Yeah.
We have time for another question.
Yeah.
You mentioned that the Phytobiomes Alliance is a pre-competitive organization. What do you mean by that?
It means that that's where we can work with industry, and I'll let Kelly jump in here because she's more of an expert in that arena. But it means that it's the research that the companies are willing to share with us and each other before it becomes competitive. Kelly, did I summarize that well enough?
Yes, our focus is primarily on pre-competitive. That's about the only place we could hope to get the companies to share data, and we're really working on that, working to try to get to that point. We have not achieved that yet. We were close just before Bayer sold all of its microbial genomes. We almost had them to be able to bring them into the public domain, but we were unable to. But one of the things we're exploring, and this is where data and how you manage data becomes very important, is can we set up these a database that allows for different levels of access? And so encourage a company to put all of their data in there, but tell us who has access. And then to also figure out a way, can we anonymize the data so that we protect either the person's farm that the data came from, or maybe even we provide some protection to IP.
And I think having a group such as the Phytobiomes Alliance, which is neutral, is very helpful in these negotiations.
Annarita, do we need to move on?
I think we have time for another short question.
Another short question?
Kelly and I never give short answers.
Pardon? What did you say, Jan?
I said we never give short answers.
No, I'm sorry.
What has been your greatest challenge so far in trying to organize all that?
I will say and I'll let Kelly take this, from the scientific point of view, is getting the different groups to let go of, so for example, we don't care who gets credit in the alliance or in the Phytobiomes world. We just want the work done. But we have trouble getting people to work together to solve these problems and to think broadly. I think that's less true now than it was 10 years ago when we started moving down this path. And I think now we're seeing that people recognize that we have to work as teams. When people were thinking in the linear way, we were less collaborative and work together less well. But I think work has shifted a lot because of the need in the gap. Kelly, what would you say would be the biggest problem that we've had in pulling the alliance together?
I think reinventing the wheel is one of the largest challenges for people, and where we see the same research experiment being done in different places, and both scientists have no idea that it's going on, or and even in reporting, and that's one of the issues, that standards becomes critical. So maybe there might be a slight difference, but we can't compare those results. But every week I get a request, or someone will say, I'm looking for a partner who can do this, and then lo and behold, we find we've got competing projects. But part of what we're trying to do is to bring those together and get them to work together. And often, here in the United States, we don't know And it's just because you're busy, you don't know what's going on in Australia or the project that's in Australia. And that's one of the things we're trying to get people to report on and to put it on our website and try to bring that together. But that gets to scientists working with each other. And then I think the other aspect is not even letting go of data. It's the whole idea of publishing under a consortia and how that actually works. And there are models for that. We've done it. We had 200 authors on the International Wheat Genome Sequencing Consortium reference genome paper, and we figured out a way that it can be cited as the consortium, but then also give proper credit to the people. But I think those require making decisions up front so you don't have those longer term problems.
Thank you, Kelly.
All right, yeah, thanks, David.
I encourage you to reach out to Jan. She'll be here at least for a day to collaborate.
Thank you. So welcome to the working group sessions today. So in the morning, we will have reports from 5 current working groups, and there will be 5 speakers, and each speaker will have 15 minutes, 10 minutes for the presentation and 5 minutes for the Q&A sessions. So we'll start with data reuse. Alenka Hafner from Penn State University, data reuse working group, first steps and goals. Hopefully, no, yeah, or that. So you don't turn off. I did put it on already. Yeah.
Hi everyone, it's my pleasure to open the working group reports. I'm Alenka Hafner. I'm a PhD student at Penn State University, and I'm part of this quite young working group that started this year. It's championed by James Coltis, who I think is on Zoom right now from Iowa State University, and I'm the co-chair giving you the report. Like I said, we're quite young, so this is only a first steps and sort of goals, and we're really looking forward to any sort of feedback and ideas we might have about the future of the working group. First, a little bit of background about how this got started, the motivation in the beginnings. So it started as an Ag2 PI seed grant, and that's because the community, and really championed by James Coltis, like I said, was concerned about the exponential growth of publicly available genomics and epigenomics data sets. And I think all of us here at the consortium are concerned about that in one way or another. But in terms of reuse, we know that data is often underutilized due to insufficient metadata and other challenges related to fair uses. And different groups in the consortium deal with this in different ways. But we really focused on reuse. So we found that intermediate details from supplemental files and also initial data and intermediate steps are not frequently captured and are not in a standard format that can then be reused. And so the objective of that seed grant was to identify bottlenecks in data use and critical needs in order to propose solutions. And there's this really nice scheme that James made here on the left about the workflow that we proposed. And we had a great online workshop last year. We had a few talks about different aspects of reuse, sort of practical and broader solutions. We had a PAG workshop this year that was very helpful. The discussion from that really started the AgBioData working group. And where we are right now is there at the end in the green and the pink. So we're working on the survey of data reuse needs and writing a white paper. So that's how we partnered out with AgBioData. Like I said, we only just started a few months ago, but I think we've made quite a bit of progress in only, 3 months since we started. So our general goals are, like I said, to identify bottlenecks in data reuse. And we mostly focus on reuse in livestock and plant communities because that's where we're from. And we're hoping to base all of those on discussions and the community survey that we're working on. We want to identify critical needs of the community to help improve data reuse and promote more fair data, hopefully in partnership with other working groups as well. This will identify missed opportunities to improve data sharing. And like I said, we're working on a white paper currently. And I'll briefly tell you where we are right now. So these are our current members. As you can see, we're at very different stages of our careers. So I'm a PhD candidate, but we have postdocs, we have professors, we also have people from industry. And we are diverse in terms of what we study, plants and livestock. And we welcome anyone from those spheres who would like to join, because it's always nice to have more perspectives. We're also mostly from the US, but we have international perspectives, even though we focus on sort of US-based recommendations for now. So the white paper draft sections, which we've written so far, are as follows. I put these up so that if you see any glaring opportunity that we missed, you yell at us, and we add it. So we've identified the major barriers, risks, and limitations to the use that we, as a quite small working group, see in our day-to-day reuse. So that would be related data standards in terms of quality and also experimental standards. Like I mentioned, the major barrier is metadata insufficient standards, and also we want to address ontologies. There's data interoperability in terms of integration, data ownership and propriety, which is why it's always nice to have people from industry to chime in. One thing that we often forget, maybe working in the bioinformatics or computational biology, is user skill level as a barrier to reuse. So we're trying to address that a little bit, as maybe the average user does not have the bioinformatics knowledge that everyone in the working group, for example, will. There's also resource availability, which is also related to point number 6, which is equity and inclusion in data reuse. And we want to discuss that further. Data availability, of course, I think we all in this consortium deal with that problem. And then, even though we focus mostly on sequencing data for now in this white paper, we're hoping to address other and emerging data types and technologies soon. As I said, this started as an active PI grant, and we're hoping that the genotype to phenotype won't be lost, even though we're focusing on sequencing data for now. So one way that we're directing this white paper draft, as well as the goals of our working group, is through a community survey. We're currently seeking IRB approval and sending it to our respective communities. And one question that our working group has for the community here is whether you see any missing link in the survey that I'll show you right now, any question that we missed that you would like to add. So there's just some basic demographic data that we have, that we ask people about what organism they work for and whether industry and academia, so forth. And then we get right into the data reuse aspect, which we know is a limited definition, but it will make the survey have more meaningful results if we define it as data produced by researchers outside of your research group, excluding publicly available reference genomes. So we asked people if they produced data before. Firstly, they said no. We asked them why not. However, we expect most people will say yes. And so we ask them how often they do this. This is quite a subjective question. We just want to gauge what type of sort of data scientists we're dealing with here. Then we ask what reuse objectives one might be interested in. This is obviously a limited list, so every question has another please specify option to it. And this presentation and the survey is available to anyone if they want to read it in more detail, we'd really appreciate it. We asked what types of data people ever used in terms of, as you see here, whether it's mostly sequencing and other types, and what types of data files they mostly use. So these are just the most common ones we were able to come up with. Again, we have another please specify option. One question that we really hope will give us an insight into what stops reuse is what would increase people's confidence in reusing data. So why are they hesitant? Why do they not reuse data if it is available? And then there's also the flip side of things. So you're not only the user, you're the producer of data. And what challenges and obstacles are making you not maybe make that data available for everyone to use? And then, although this isn't a major focus of our paper, we do want to ask people if they've reproduced other people's data, if they've tried to do that, as that is a type of reuse. And just two questions, whether they tried and if they were successful in doing so. And if there's anything else they'd like us to know or address as a working group. So if you have any additions to this, if there's anything we missed, we would love to know. In terms of future directions of our working group, we, like I said, are focusing on sequencing data for now, but want to address emerging data types and methods in the next stage of the working group. One thing that we're quite interested in the long term is machine learning assisted metadata curation, as we think that would solve a lot of the problems and barriers that we're having in terms of data reuse. And obviously any other ideas that we can come up with today in the breakout session. So please, if you have any ideas and maybe you can join the breakout room or just had a whiteboard go off now. If you scan this, it will go to a Google Doc where you can jot down any ideas about the survey or in general about the future of the group. Or contact me and James at our emails here, but we're hoping that it will sort of be a brain dump on this Google Doc that we can use. So thank you so much.
Thank you. We have time for one question, one quick question. So I think so. So hi, thank you for your presentation, Alenka. So I have one comment for the survey, like when you ask if people reuse or not data, I don't know if you mentioned to ask if people don't reuse data, why? Like I guess, like I have been being a data generator for many years, and sometimes you cannot find the data or you don't know that data are already there. I don't know if you are already exploring that side, that if you want to see that perspective and this way.
Yeah, definitely. So this is the options that we had in the why don't you reuse data. But we are potentially looking at expanding this section because this is the only question for the people who have not reused data before. So yeah.
Thank you. Yeah, so next speaker is Annarita. So diversity, equity, and inclusion. Yeah. How do we address all the aspects of DEI in AgBioData?
I hope you can hear me. My presentation. Yeah, so I'm also the chair of the recruiting working group. So there is not so much about genomic or phenotypic data in this working group, but what we do is trying to expand our network and try to involve as much as we can new members from different fields. So I want first give an overview of who we are. We are a very great group. We had great discussion in the last 2 years. So there is me, that I'm the chair, Madam is Annarita, Rosa, John, Neil, Margaret, Chu, and recently also Susan, she joined us, and she's the champion of the working group, and she's bringing insider experience in the field that we need for improving. So as other working groups, like the education working group and the sustainability working group, we are one of the 4 core working group of the grant. In particular, we are around 82. So we work first to recruit new members for the working groups. So even any of your working group needs new members or anything, you can reach out to us and we will try to help you. But also we try to recruit new members for the entire network. And we try to, we develop outreach resources and material to teach people about who we are, what we do, and what is our mission. And so when we talk about diversity and inclusion, we are talking about different levels. For sure, we want to involve key stakeholders that are part of the agricultural research. And so we are talking about, for sure, data generators, bioinnovators, librarian, publisher, anybody that use or reuse or manage data that can provide insight and different perspective about the data, right? So I guess the perspective of the data generator, submitters is different from the biocurator. We also try to, we specifically target particular communities that are underrepresented in the field. But also we try to empower early career researchers, trying to involve them in the network and be part of and try to use the advantage of this community. And we also are starting to think about scientists from less well-funded institutions, both domestic and international. And our recruiting also towards databases resources that they are not part of the community, the consortium yet. They are probably representing small communities in the research field, but we need to acknowledge them and to involve them in our consortium. And try to embrace database that reflect the diversity of organisms used in agricultural research. So here there is this amazing thoughts that I have to give the thanks to Megan or Bennett that she's there. She's from the Kitchen Working Group. She generated these plots for one poster about the Education Working Group, which we are part of. So here there is kind of a brief description of the diversity in terms of job titles of the working group members. So you see we are quite diverse. We have early career researchers, postdoc, graduate students, a little bit of undergrads, but we also have data curators and people from different type of institutions. Most of us are from higher education institutions, but we also have people from government, research institute, non-profit organization, publishing, and industry. But what these data don't say is that the majority of the scientists that are part of our community are plant scientists, and plants are not the only, crops are not the only part of agriculture. So we are really looking forward to involve more people using other organisms like animal scientists or insects. So if there is anybody there in the community that we're also virtually that joining us and hearing us, please reach out to us, and we would really love to have you because we think that you have a different perspective on data that we need to hear from. So in terms of targeting underrepresented communities, so this is an action that Marette and John work on, and recently Susan, she brought some insight about it. So we thought that, first of all, we should know about which kind of HP, historical Black college, university, and minor setting institutions are there with agricultural programs. So they work on a list of these institutions. And then we decided to email them and invite them for an informal meeting where we can present who we are, what we do, and then hear what is their experience with data. And we get almost no response. Which is something that is like, we are still working on it. But this raises another important question is like, why people in general are not interested in joining AgBioData? What really
them to get interested and actively participating, because the AgBioData network is very large, has many members, but a few participate in the working groups or generally in our activity. And so what is like really limiting them in getting interested? And so regarding the first challenges in why they didn't answer, so we should think about what didn't work about this approach. Maybe we are using an approach that is not very effective. What should we improve? And this actually comes to, this is something that Crystal Sosi for our last webinar, it was an incredible webinar where she talked about the insurance care principle. And it made me think that most of the time, these underrepresented communities, they are overwhelmed about this email telling them, hey, come join our community because we really want to hear about you. But probably they have other type of issue that they are already trying to fix. Other perspective, like as indigenous people have already done, we already say like, hey, these are our data, we want to protect them and we will meet our principles. So maybe we should approach them in a different way that we don't know, and maybe we should think about that. And also we had like a lot of discussion about what does it mean participating in AgBioData, what are the advantages of being part of AgBioData. And so what AgBioData especially can offer to you as a scientist. So we listed some of the advantages that are already part of AgBioData. For sure, when you are part of AgBioData, you have the possibility of networking, learn new skills, learn new tools. We provide educational material that will be accessible for everybody, but also we do monthly webinars where you know about people and resources. But still, we think it's not enough, and so maybe we can work about something else. And these were some of the suggestions that the member of the working group, they have. And there is, for example, money. And this was somebody of the working group that told me, like, look, if somebody comes to me and from my community and say, oh, do you want to be part of AgBioData? They will ask, is there any money involved? And you say like, but that's kind of, it's not, because as you all know, most of the time that you dedicate to AgBioData is volunteer job that you have to take off from your work for which you are paid about. So I also participated in another RCN grant where they actually gave me money, and that motivated me to participate more because I was kind of feeling responsible. But this also brought me to learn a lot about that RCN project. So maybe this can also be something that we should think about in the future in grants. We also think that we can offer a mentoring program. This community involves many different professional areas. There are biotraders, librarians, people working in government agency, faculty. So maybe we can provide early career research a different pathway advice about how you can develop. Like, I didn't know how to be a biocurator, so what do I need to do? What kind of skills do I need to be a biocurator? So maybe we can provide that. We can provide support with data management plans. Most agencies now, they request about that. And maybe we can provide guidelines suggestion on how they can do that when they are preparing grants. And this was another suggestion, like consulting for grant. And I hope there will be insight in the breakout room about this kind of insight, also based on your experience in participating in AgBioData. Will this motivate you more to be part of working group or to actually participate in our consortium? And please, the people online, give us any feedback. So I want also to give some achievement, they are not only challenge. So since the beginning of the grant, more than 100 people have joined our community, and we are very happy to have them with us. We have also involved and contacting new database resources, micropublication, Ensemble, and European Variation Archive. They joined our consortium, and we also had contacted with Grapedia that we hope will be part of the consortium soon. So we have a plan that to reach out to underrepresented communities, but we hope that after this meeting, we will have new suggestions to reevaluate this action and be more effective. And also we invited key speakers for the monthly webinar.
So what we will do this year, this working group will last till the end of the grant, so we have a lot to do still. And so we will also try to explore new services that AgBioData can offer and try to involve the community in that. We will reach out to new databases resources, so please, if you know any database that is not part of the consortium that we would love to be part of, so reach out to us. And maybe we can combine the educational training with outreach to involve more underrepresented communities. And so I want just to recap the question that we will discuss during the breakout session. So what are the most effective ways to engage underrepresented groups in AgBioData? How can AgBioData best serve the needs of our diverse communities according to the different stakeholders? And how are there any other data management groups that we should contact? And with this, I want to thank the current members, but also past members that they were with us last year and they left, like Doreen, Adrian, Erin, and Jacqueline. And of course, thank you for listening.
We have time for one question.
So, both of these recruiting and data reuse, there's a couple of really successful examples that a lot of us wouldn't consider. They're not genomic data, but the National Research Council's animal requirements has been around for about 130 years. And they are required by statute and funding to share data, and then it goes out to the entire industry, and it's funded by industry and it's funded by government. Another one is the entire animal gene or bull proofs and sire proof system. So a lot of, you know, as we focus on genomics and genetics and phenomics, we kind of forget those were our database management systems with a very successful track record. And I think that can merge into this one with education. Mm-hmm. You know, what's out there for both students, not so much will you join our group and share data, but how can we educate your students for careers, that type of thing.
Yeah, thank you. Yeah, so next speaker is Jacqueline Campbell, USCIRS scientist, and she will be talking on fair scientific literature. Thank you.
Hello, let me know if I talk too loud. I have a booming voice, so if I'm too loud, you know, let me know. Hi. Sorry, everybody online. Lenore. We'll have a conversation later. All right, so I'm one of the members of this group, and I'm actually really excited about being in this group because it's one of those things where I feel like I'm passionate about this, like it's just the driving force. So what are our goals? Well, one is like finding the bottlenecks, like what is the problem here? Trying to find what is the bottleneck between the publication and the curation pipeline. Identify sets of allergies and invoices. Identify sets of desired or existing tools that can help us with this work to really increase the curation resources and the accuracy. And then really to the second push next year, second year, publish recommendations for a roadmap for authors and publishers and other stakeholders to really increase the fairness of published research. So here are our members. We actually have a wide diversity here of curators like myself, computational biologists and professors, and even some members from the publishing community. Where's Megan? Megan, I think we should drop in every once in a while since you're our librarian. And the other librarians here too, sorry. I just know Megan's really well. So, our main, like just thinking about, you know, who are our stakeholders? Well, we think about, okay, we are a stakeholder as databases. So here's the first, okay. Genetic genomic breeding databases or genetic genomic breeding curators like myself. What do we do? We make data findable, accessible, interoperable, and reusable. Right. So here are currently our, we want to get from the author to our databases. We want that data in our databases because we're trying to make it fair. So what are our challenges during this whole process? And the stop signs. So first of all, a high volume of data, and there's not enough time to do it. Really, like there's a paper coming out as I'm speaking right now that I should be reading. And then, of course, difficult to find the papers that actually have relevant data that I can even use in the first place. Reading a paper, you go, oh, this is great, and no, no way. And then the classic, you ask for data on request, you get nothing back. I have a general rule, if you don't email me back in 72 hours, you give me the right to email bomb you for a week. It does not work. I tried it for 2 months, it didn't work. Last thing, poorly or incorrectly formatted data and metadata, bane of my existence. I read a paper, great, data is available. It'll take me a week to get it formatted correctly. And then finally, yes, yes, it's in the database. And whole process probably a month or 1 week. No. So we're aiming for a better workflow. All these stop signs are really hampering our work. So first think about like who's our stakeholders, who are we involved with? So we kind of went through this whole group and we called it the personas table. I can share it with you later if you want to. Think about all the stakeholders that are involved in this whole process. Who is interested in the data? Who wants the data? Who is messing with the data? What do they do with this? And we have a large variety of people here, but we're going to focus on 4 because we need to focus. Focus is saying no. So you're saying no to the others. Sorry, next. So focusing on these 4. So first, okay, what are the challenges? What are the barriers that they're having? Just like I described the barriers that I as a creator are having. Oh, you're laughing, Jennifer, you left that, but you're like, don't. And I don't know if you've noticed, the top one in every single person is highlighted because it's exactly the same. Not knowing where the data should go. Okay. Then what incentives, what resources could we generate or give to kind of this? Again, something that comes up often is resources to determine where data should go. Okay, well, that should be or resources for tracking where data goes, or tools for sharing or even uploading. So we really could be the bridge to make this a little bit easier. I mean, you do not build a bridge in like 2 years. I mean, it takes like many years. Rome was not built today. So here's our questions for the community. Is there a tool or portal that really can help our stakeholders to determine where data should go? And if you're thinking about it, I mean, the imagination is go for it. What would it look like? What would you want? Like dream big. Really want you to like dream big. Thinking of the barriers and incentives. What would make it fair for data? Like, are we missing something? Is like we think out of the box, but we can only think out of the box for like within our head. Someone else has a better idea usually. I like to crack their head open and figure out what that idea is. And last but not least, what are the most important challenges in this process that we are missing? Right? So that's all I have. Do I have extra time or it's done? I have done. Okay. I want to tell a fun story because how do you do it, Leonore? Archimedes was a wonderful advanced mathematician in Rome. And all his documentation was there. But he, of course, he was told when Rome was sacked, the soldiers were told, I think, well, he insulted the soldier because he was busy fighting on his man, so killed him. Great. Well, all his documents were lost. There were 3 of them, 3 books. Okay, it was lost, and like during World War I, he was lost. The document, I can't remember when, but it was lost. And then they found C. It was written over by some monk because he needed paper. And the mathematician in the documentary was saying, if we found this beforehand, if we knew about all this math, we would have been 100, 200, maybe 500 years more advanced in our math than we are now. Just because we lost this data, we're behind. I was just like, why I'm passionate about curating data, because if we lose this data, we are now scientifically, mathematically, society-like behind. So I finally get to tell that story. I love that story. Any questions for me?
Yeah, we have actually time for 2 questions.
What then?
So one of the things I've been looking at in some of the other work I do, and I don't know if this is something you guys have looked at, is in terms of the pipeline, getting data into the database, is getting data into a database as soon as it's collected. So I've been working with the KSU Field Book group. So as soon as they collect data in the field, it's dumped into a database. So that whole process of the curation is 5 steps ahead, just because it's already there, and then they can do their publications on it, whatever they need to do from there. So is that something you guys have looked at in the working group, or something you've explored?
In this particular working group, I don't think we've explored it, but I know within our database, when we've talked about it, there were 2 main issues. One, integrating software together, interoperability. That's always become an issue. And 2 is the fear of being scooped. Like if you put this in a database, it's someone else could see it and like, oh, they're gonna write a paper faster than me because you go on maternity leave or something happens. So we have something like that available for some, like it needs to be password protected in some way or fashion. You have to advertise that very much, that your data, if you enter it early, is password protected.
Well, the other issue on that is it's not peer reviewed. Yeah. And you run into a really, really serious issue with that. But again, overarching to all of these, and you talked about it, but the big question is who wants it? Mm-hmm. Who wants the data? I mean, I will agree with you about data loss, clearly, but people who have billions of dollars to spend may not share that same concern. Yeah. So who wants that data? Who is going to build, pay for a sustainable system? That's the bold, right? That's the out of the box kind of thing we need to talk about.
That is a good point. And we try to make some assumptions that other researchers want this data. And so, I mean, that's a question maybe we just ask more of our own individual stakeholders. Like, why do you want this data? Why are you coming to surveys? Or why I work for surveys? So why are you coming here? That could be good. Another question?
Not a question, but it's a follow-up to yours, Peter, which is data release upon publication. There's another resource that we work with called Morphobank, where researchers can collaborate pre-publication, collect their data, and they only have access to it during the data collection phase. And then when they publish, they can set it to public. And during the review process, they can have anonymous reviewer access to it. So it's been set up, it's expensive to set up in the first place, but that's a way to get it, like collecting the data at the point, and then you push button and you release.
Before you send me that info, please, you just mentioned, make it cover.
Okay, so I'm a librarian and I manage a generalist repository for our school. And I can tell you, publishers have absolutely zero workload on repositories right now and databases. And it's been a lot of fun trying to contact them and they just go, no, this is a press release. I'm like, I don't want your press release date. I need to know when it's published, I can release the data set in the BOI the same day. I mean, literally do not know what to do with something to the press people. And I give up. This has happened multiple times. So like the number one thing is like, we need the publishers to be like, yes, there is a workflow for this, and we need to talk to the repositories and databases like they're equals.
Yeah, we were talking about incentives to publishers. So like, for example, that UI that you would put out after it's an incentive to them. If you kind of like sell it, it's like selling data, like, hey, yeah, this is worth some money. Then they're more interested, I think.
Yes, I agree with her. I think it's especially the small journals that they are from societies. It's a big journal. No, I think there would be like involving more publisher on our biodata. Yeah.
Great ideas that we could talk about in the breakout rooms.
Yeah.
Breakout rooms.
We can actually we can have one more question.
Oh, really?
Yes.
Karen, she was
Yeah. Then do you want to?
Turn.
So, yeah, along these lines, I was thinking AgBioData could be that voice that does talk about the data. But I also plugging my publications how they talk through and come from a database and want authors to give us their data rather than curious for the finding. So we incentivize authors to research to give us their data by publishing. So we can board that judge citation as well as it is peer reviewed. So I think this is a good model going forward and so
Thank you. Thank you very much. The next speaker is Marcela from Cold Spring Harbor, and she's going to talk on standards for genetic variation. Thank you, Marcela.
Thank you, Sunita. Sunita and I work under the supervision of Doreen Ware, who's late for the meeting, but this afternoon. Okay. That's, oh, not good. Oh, okay. So this is what I'm going to tell you. First, I'm going to tell you we are. Then I'm going to tell you what do we want. And then it's going to be a quiz so you fill out what we have done in our products, what challenges we have identified, and then how we are working towards coming up with solutions. So this is our just a representative meeting. This is one where we have a few more than usual. Our group is actually not that small. It's we have over 20 people, I think. But again, usually we're not everybody's involved, but we have a good representation. We have a representative biocreators from Green, USDA, and community databases and other. Okay. So our goals. First, we want to identify practical challenges associated with sharing and reusing genetic variation data sets. Second, bring together a community of data providers, biocurators, and computer scientists to promote interoperability and access to those data sets. Third, support the harmonization and adoption of those standards from various platforms in both plants and animals. What we have done, we have monthly meetings. We have had biocreator discussions. We have set out surveys. We had a webinar with short talks on data providers as well as representatives of platforms that handle genetic variation and breeding data. And then we are working with pilot studies, biocurating data sets. So far, we have identified what is critical and desirable information from germplasm curators for those genetic variation sets. Second, we have identified practical challenges associated with sharing and reusing. So our goal one is check. And we are working slowly towards the verification of those data sets without pilots. And I'm going to tell you a little bit more about each one of those activities. First, you might have heard about these. We got a group in Europe gave us a head start because in early last year, they came up with this publication with recommendations for BCF, the standard format for genetic variation, and they have clear guidelines, checklists for, especially for metadata. And we're lucky that 3 of the authors in this paper are very active members of our group. Sebastian, Sarah, and Tim, who is the co-chair. So this is the table 1 from that paper, and if you look at the sample, there is a field for samples, the very specification of what they're looking for, including, an identifier. We want 2 things: identifiers for both markers and identifiers for samples. And so this is focused on the sample. So the idea is to have, in this paper, is to have biosample IDs. But we realized during our discussions with the biocurators that it's very important to have germplasm IDs. So we can be linked, those can be linked. Otherwise, it's very hard to identify what, a common sample, that biosample ID, which is a little obscure, belongs to. So, yep. And this is from our working meeting, we came up with additional suggestions. But again, these might not fit super well. So those metadata recommendations are really for the BCF file itself. And when you have like a huge amount of samples, or it may be complicated to have that many lines, commented lines for metadata. So we have been discussing that it would be best to have that information in the biosample entry. And that's kind of hard to incentivize data providers to play in those fields, but it's very, very, very important for us to be able to connect and facilitate interoperability. So yes, so we advocate for requiring a primary external identifier from a major germplasm repository with a GUI for that particular sample. And in many cases, like in green, the green, a PI number in green, you might have seen it has a lot of information, passport information, pedigree information, and that can be linked to other resources. For biocurators, it will be useful, but not essential to have inventory, local number, that kind of thing. So that was one of our achievements. Surveys, I'm going to tell you quickly. This is one survey that was run among us for the bio resources. And I'm going to tell you 4 key findings. So you can see on the first one, the question is, how do you share or plan to share your variation data? Almost all data sets are publicly available, but formats and methods for sharing are very diverse. Only 33% deposit to central databases like the European Variation Archive. So that means that the other 70% is not, and that's complicated. Some of them might go to species community databases, but it's part of the UC a little more later. Second question is the variation data linked to other accession data, like for example the biosamples? Cross-linked into important metadata does not always happen. 40% responded that they do not link to even the reference gene. So that's a very big challenge for us. Third question, are the stable variant identifiers associated with the variation data that you hold? The majority do not use stable variant identifier, like RSFP. If the reference genome changes, how do you handle this aspect? When new genome is available, about 1 in 3 respondents rematch the raw data. About half do not update. And if you want to see all of the results, there's a link on the slide, which will become available to everyone. So for the webinar that we held, I believe that's December, I told you we had 8 great presentations, different resources, very, it's a good representation of AgBioData resources. Very little from animals, but we did have. Outcomes, we identified variation data sets in a wide range of species. We sample diversity in data submission, informative, in processing, display and analysis tools, interoperability, and in different use cases, which was nice. And then we also recruited new members from those presentations. Our pilots, I'm going to take, okay, so just to give you a quick, we have to strategize how to choose which studies to use to sample, to identify challenges. So we try to go with species that have, it was the first, the top ideal is high quality reference assembly that are registered in any of the INSBC databases. If you're not familiar, one is ENA, European Nucleotide Variation, the other is GenBank, and the other is the Japanese database. And that they have genetic variation data in one of our communities databases. Second, especially with, again, a high quality reference assembly that is not submitted to INSBC, but the variation sets do not have resources to host large data sets like green, for example, or the flowers. And then we have high quality references assemblies, but are developing new data. Okay. And this is more or less where we stand. So with green, I have checked those things that are available there. With orange, it's very hard to get that information. It's not there, and it'll be very, very hard. It will depend of the authors most often to have that, make that data available. Yellow is something that we are working towards achieving. And just to mention some, sorghum is this study, Lucas took part of in our discussions, right? Discussions. So the sorghum assembly is registered. He provided the DCF, made it available. He submitted his data to the EVA. So EVA is in the process of assigning RSIDs to that data, and also one thing our blog right now is to have that those the biosample entries do not have are not linked to PI, but the information is there. It just need to be a little bit of work. So that's something we are working on, so that's the most progress we have. That's our start trial of all the time. Then we have the worst case scenario where there is no reference assemblies, like several of the fruits we have numbers from the NCPR resources, and there are some where the species is not even, the assembly is not even a DNA. So that's a big challenge. And then we have other data sets like grape or maize, where there is a new publication just came out, and it's like, supersedes many others because it's like the newest, latest methodology, and it's a good sampling, and we have everything. But and also we have a high reference assembly in the ENA, and we have a community database. In this case, one that I work on, Gamine grape or Gamine maize, that is interested in following up and getting that information. And so we're pursuing, we're coordinating with whoever we need to. For maize, so maybe in the next one, I'll give you those examples. Yeah, let me just move. But just so you know, here is there is a coordination, a high level of coordination needs to happen between the database who's gonna host the genetic variation between the EVA, between the data submitters to have everything. So far, this is a summary. We review guidelines and propose additional recommendations to support adoption. We identify data sets, workflows, and technical barriers. And the challenges that we discovered through the exercises are listed as missing reference genome. Reference genome is not registered. Because you might know, some of the variation sets do not even have a reference assembly. They use some other species as reference. That's what the first one. The first two, the other is there may be a high quality assembly, but it's not registered at the NDC. It might be stored somewhere in a community database like DBR, or but it's not. We cannot reuse it. And one of the requirements to submit your data to EVA is to have it in at ENA. Variation is not readily available. It can in the publication, it will appear as something like request from authors, available through or a private FTP, and so we cannot. It is not meant to be really published publicly, or if there's some agreement. Variation is not in a standardized format, yeah, and some of the the things that I discovered with these biocuration exercises were like, the genome might be at DNA, but they only will make reads, the sequencing reads available. So most biologists are not going to be able to, ingest that data or take that data, reuse the data. Other things that I am not a computer scientist, so I want to be to learn about genomics, data stores. And I mean, it is great what you can do with what I read that you can do with this, but most biologists are not going to be able to do that. To generate PCF files from with the tools in there, it sounds great, but it's a challenge. And then or it's not going to be PCF, it's going to be just an Excel file with the variant from some like phenotyping array. That's it. So those are some of the challenges that we discovered and how we work into our solution. We have several of those challenges. Here are some of the proposed solutions, ideas that have come up so far. We don't want to focus just on how do you say, punish, people who not share the data, but how do we integrate the people, right? Like it's been mentioned before. The carrot and stick model. We have also, there is other project like the parent project in animals that is, we're trying to come up like members from our group, including Doreen, Sarah, having in conversations with like Chris Gardler, with Peter Harrison for a plant bank. And that's to reuse the tools that they have, so we can, have those biosamples, acquire that metadata, that type of thing. So these are just a few ideas. And from hearing the talk as the speakers earlier, I thought about like, okay, we really need education and training. It's not only people don't want to submit because it's too complicated to submit to GenBank, and so come out with easy guides, and Elixir had made like a really good documentation for that, like they have a cookbook for submitting that, your assembly. So, but people don't know. So we have to make that data available, that information available and incentivized. So those are just some ideas, and I'm not going more detail. Thank you, and I'm just gonna leave you with the breakout group questions. If you're interested in joining us, very welcome.
Thank you, Marcela. So there's no time for questions.
We have one question.
One quick question.
So in order to generate the VCF file, you have to run a bioinformatics program to align raw reads against genome sequence and call SNPs. So is there, are you also recommending a standard best practice for that pipeline? Because I can imagine, that's also important metadata for you to provide in the BCF file, and also it could determine the quality of those SNPs as well, and then that should be known when you submit it to ENA, right?
Yeah. Right now, we have been only focusing because of the nature of the working group. We have been focusing on the biosamples, capturing and metadata, but it is very important. We should be considering that might be something that the group that focus on the BCF could do. But yes, that's a great suggestion for Team Center when they are capturing that metadata for the entire documents.
Thank you, Marcela. The next speaker is Shabari Subramaniam from Phoenix Bioinformatics, and he is going to talk on sustainability. Thank you.
Thank you.
It's nice to be here, and I want to start off by thanking Leonore and Annarita for all their massive effort. They were putting this together, and it's really nice to see you all. Those of you who are part of the sustainability workshop or working group, I get to talk to you often, but it's always good to share these ideas with the general audience so that we can get everyone's views incorporated. I'm part of the sustainability working group for AgBioData, and part of this RCN. As Leonore had mentioned, we've done some work on this, on sustainability as part of the year one work. And the working group itself was put together very recently. And the goals is to then take what we've done in the first year and build on it. And then hopefully at the very end, come up with some recommendations for sustainability models that would help for the various AgBioData resources. So the goals of this RPN as far as sustainability is concerned is we want to do a self-assessment of the long-term financial stability of the various resources. And to do this, we want to do an analysis of all the modeling solutions, modeling of the sustainability solutions for a representative group of AgBio databases. You have about 40 databases here. And when we looked at the various databases, the wide range of not just the organisms, but the data types and the size of the research community, the type of research community, and so on and so forth. So, which is great. Lots and lots of very interesting, very diverse sets of data sets for us to play with. And it also gives us an opportunity to test out different types of sustainability models. And the end goal, of course, is to work through years 2 and 3 of this RCN grant to eventually come up with a roadmap for database sustainability. And as the first year plan, what we did was we reached out to the principal investigators of various resources to try and get an idea of what the resources are. What are these databases? What do you have? Which are the organisms that you're supporting? What is the size of your database? What's your employee strength? All that sort of thing. And so we did that through interviews as well as manually surveys. And what we did was we sent out, we created surveys with a SurveyMonkey, and we sent out links to the various AgBioData database PIs, got feedback from them for various questions, and then we reviewed those questions, we reviewed their answers to try and identify various aspects of it. The first thing we want to do, typically when we look at a resource, is to identify who are the stakeholders, who are the folks who find this resource absolutely invaluable. Because those are the folks you want to be in the room, to really be our champions, to go out there and talk about our resource. Because if you're not doing active active research in a particular organism, chances are your interest in the sustainability of the organism is a little bit more diminished than someone who thinks it's absolutely vital. So, and so the first thing is, of course, to identify folks. And it's easy to assume that, well, it's a database, so it's pretty much the same type of stakeholders. Not really. So, so we reached out to the PIs to kind of understand from the PIs who are the folks who really value the resources. And so we collected that information from them. The idea behind that is that then helps us with the second stage, which is now we get to reach out to these stakeholders to try and understand how much you value them. See, this is, you know, some of you are, most of you are PIs of these resources, and you guys talk to your community members, so you have an understanding of how they use it, how they would like to see this resource improve. And so when you talk to the folks themselves who use these resources, then you get a different perspective, because it's hardly ever that as PIs you ever have a sustainability conversation with your users, and you're always going to be about utilities. Like, how can I make my resource more wonderful? Right? Now, what data sets would you like to see that? And that is an important conversation. But the sustainability conversation needs to come in where folks need to realize that this stuff ain't free, right? And it ain't cheap. So they need to know that they have to play an active role in helping to keep this resource, these resources running. And so this is just a summary of some of the things we found out, not very surprising. Most of the stakeholders in terms of users, according to the PIs themselves, graduate students, academic nonprofit researchers, for-profit companies, great, breeders, and others. And this helps us because when we then build our next set of surveys to reach out to these stakeholders, we can then have a whole different, a diverse set of stakeholders to reach out to. So it's not just all, you know, academic researchers, for example. The other thing, of course, the key aspect is to understand how desperate is your situation. Because, you know, that's kind of why we're here, right? We talk about, ideally, we'd all like to have everybody funded, awesome, okay. But we want to know that we do know that some of the resources are far more insecure, financially speaking, than others. We want to understand what's your situation like. And that helps us modulate that conversation with your users. Because it's highly unlikely that all your users understand the level of financial security or insecurity that your resource is having. And the other thing, of course, is to understand how do you anticipate your expense of changing over the next few years. Because it's one thing to talk about where we are now. Some of these resources, a lot of these resources are still funded. And that's when you want to talk about sustainability. You don't want to start thinking sustainability once you run out of funding. You want to start thinking sustainability when you are still funded. Right? And you look ahead and you think, oh, I have 2 or 3 more years of funding. Let me think ahead and plan towards it. The other key aspect is data persistence, which is people don't realize these resources hold valuable amounts of data that's been collected over years and years and years. What happens to that data if the database disappears? That's not a question researchers ask themselves. They think, oh, it's there, it's always going to be there. Right? Or it's this other wonderful resource I've used for 20 years, it's always going to be there. But this is the key question that that PIs have to ask. That you know, what happens to this data? And so that was important for us to understand also, what are the different ways in which you think your data is, what would happen to your data, you know, in the future. So I can keep going on and on, but so there are lots and lots of these questions that we have that we wanted to ask, because we want to use that as the sort of the foundation for reaching out to the communities, the stakeholders, and try and understand what was important to them. Frequency of data updates, another very important thing, which is most of you who are PIs of data resources have encountered this. People asking you about how often your data is being updated. People think data updates are really, really easy, right? But it's not. I'm in the middle of doing one. I know it's not easy. So, but it's value added, right? The reality is that we all like to keep our databases, our resources updated often, but the more updates you want, the more costlier it is, because the more people you need, more resources you need. There are several different strategies we talked about in our survey, and also asked PIs if they've considered in the past steps on how to reduce costs, for example, sharing data with other databases, things like that. User experience, this is the key aspect. We wanted to know from the PIs what they thought their community would be comfortable with that sustainability model. Would everybody be willing to pay for a subscription, for example? Or are other models things that they would be willing to consider? Usage capture mechanism. Now, this is where you as a resource has a mechanism to know who the users who come to your resource knows you, do they use it, so on and so forth. And so there's that one resource that they don't have a usage capture mechanism yet, but I'm sure they will implement that. And other more functional aspects like private data workspace, things of that nature. So the key points that we came back with the first year survey was that it's never too early to think about your future. And the unfortunate reality is that the part of the database that everybody values the most, which is the curated data sets, the ones that are at risk of the data resource disappears, right? Because the curators are not there on my curated data set. So the more value we can give it, the better. One of the key recommendations that, and this is something that I'm going to keep hounding you guys about this and the research community, the stakeholders, is we need data. If we're going to come up with effective models, we need data. So those of you who are PIs of resources, you're used to being me and Leonore coming after you the last year, pushing you to fill this array. Thank you, all of you. But your stakeholders are going to feel the pain from me because I'm going to be reaching out to them, and we need your help. We need your help to reach out to communities and help them understand the importance of providing us with this data. Right? And as suggested earlier, there are things that we have to go through. These are not just simple surveys. We have to go through IRBs and so on before we can get to that point. So those things take time. And once we get through to that point and we get to the final point of sending out these surveys, we would really appreciate the response. There are several different models we have considered and we are considering. As far as the working group specifically is concerned, we are still very, very young. And what we've had a chance to do so far is to sit down and have a conversation about which of the 40 databases do we want to pull out and use for our further survey. So we want to identify as many models as possible that would be representative. Right? To do that, we wanted to come up with a set of criterion. And so we met a few times and we've come up with a set of criterion on what you would use to decide which of these 40 databases, and on which I make at least like 6 or 7 of them, and then go through and then work them through the process and see which of these we can use for the data analysis. These are the team Phoenix. I'm not sure where my questions are, but either way, so some of the questions that we had discussed in our working group meetings are specifically to do with when we target a sustainability model for a particular resource, what are the considerations that we have to give it? And so as you come in with within the questions, we'll have more. Thank you.
Quick question.
I work for a USD database, and there's a lot of things that we cannot do, federal government regulations. So if there is another database that we know, can we give, like, they're not part of AgBioData, but can we give them their contact info and you can maybe send a survey to them, even though they're not part of this organization, because you don't want to, but I mean, what can we do to get, to help you get more data, since a lot of USA databases, like I was told I couldn't fill up that survey?
Yeah, I mean, that is a true point, but we, I don't know if it be representative of the resource that this is specific to the RCN or so it's a non-active data. I'm happy to have a conversation.
Thank you, everyone. I'm going to do report outs from our breakout rooms. We are going to start with data reuse, and Alenka is going to come give us an update on what they talked about.
Great. So I think we had a good discussion in the data reuse breakout room. We had quite a few people, 13 and 14, which was great. So the two questions that we talked about, I already mentioned in the presentation, but just to reiterate, we wanted to see whether we were missing any major issues and barriers to data reuse with our sort of white paper draft sections that I presented, which you can see here on the left. And we had a good discussion that started out with trying to get more connection with different working groups. We saw that there was a big opportunity to connect with the Fair Scientific Literature Working Group, because we're talking about some of the similar issues, but we didn't really know that we were doing that. So we're hoping to communicate with them further and address some of those links. One thing in particular is that linking biosample IDs and other information in publications is a very important barrier or opportunity for reuse. And so we want to really describe that in more detail in the paper. Although we briefly mention it in our white paper sections, data standards, particular data quality and experimental standards, were also important to the people in the breakout room. But one thing that we haven't really addressed yet is the commonalities and unique challenges of plant and animal data standards, especially when it comes to data collection. So that's not something that we were intending to really dig into, but upon discussion, it seemed like that was a big question that we need to address. The importance of getting data into a repository as soon as it's produced was mentioned before by Jackie. It was also pointed out in the working group. One thing that we discussed is that could also be a challenge, because real data going directly into a database, especially when it comes to phenomics, could be a big potential for error. And so community creation here would be important. We're not saying that we'll solve all of this with one white paper, but we will sort of discuss it as a challenge. And then finally, I briefly passed through it in the presentation, but making intermediate steps of analysis, sort of data objects, not just sort of description of a pipeline, this is what I did, but actually making those objects available and actually reusable, hopefully machine readable at some point, is sort of the, I guess, pipe dream. And on the way to there, we need to make the workflows available. And I think that was it of the major future actions we can do regarding the first question, which is really just a white paper. And then we looked at the questions on the survey that I presented before, and we're trying to see any major gaps in surveying the community. And it seemed that we sort of missed the opportunity to survey data curators. We really focused our survey in the first pass on data producers and sort of everyday users, you could say. But except of asking, are you a data creator? We didn't really go further than that. So we'll hopefully add some more questions on it. One thing we didn't ask and we will add is when trying to reuse data, did you have to contact the authors or was the data already comprehensively described? So there's that emailing of do you get answer and so forth. So that would be a good thing to know, and how many percentage of reuse cases that actually happens. Like I said, the question for curators would be, what are the most common barriers that you experience as a data curator? And that would be distinct from the question that we have now of the data reuser, what challenges they most often encounter. And we already have a question about what the barriers are to uploading data, making your data available. But we didn't specifically ask when you produced and disseminated your data, did you have the data reuse in mind? So we just asked, did you do it, but did you make the data thinking that you were going to do that? And then also we have a question already about the difficulties in submitting data that I just referenced, but they're not very technical, the options. So they're more broad, sort of what part of the process did you have trouble with, but they're not technical. So we're gonna make that a little bit more specific. One thing we briefly mentioned, didn't really have a resolution for it, but should we have a question about the incentives of data reuse? So we do have a question about objectives, but we were sort of thinking, is that the same thing? Do we need to add a question about incentives of data reuse? So yeah, I think it gives a lot to think about, both in terms of the white paper and the survey. So if anyone would like to join to further this discussion, please do. We'll love to have you. Thanks.
Thank you, Alenka. So to everybody online, we're going to do report outs from our in-person groups and not from our online groups, but don't worry, we value all of your information and discussions, and those will be given back to each working group. Yeah, Annarita might have to. So next up, we're going to do standards for genetic variation, and that will be Marcela.
No, it was me actually, but it's okay.
Oh, is that not the next one?
Well, it's okay.
Okay.
It's not the one left.
It's okay. Okay, so I was rushing getting my slides. I may not have a chance. Oh wait, no, wait. Oh, there. Can you? Oh, it's very distracting. Hide video panel. Is that it?
No, hide floating.
Floating. Sorry. Okay. Okay, the first question we had is how do we address the challenges of standardized formatting and sharing genotyping data? Because we get raw sequencing or array data are fine, but not standard BCFs. So stakeholders need to understand that samples and marker IDs are needed for others to reuse data and have your data in multiple resources. So some of the feedback we gathered was that some programs do not output the BCF. So we need converter tools, like going from Excel to BCF. We're aware that BCF validators exist. There's one IBI, there is one for the human, I don't remember, it's BCF tools, it's in BCF tools, the GitHub or whatever. Community databases could support users to validate, and we have an example, well, maybe more than one, are interested in helping just validate BCF already made, but not to actually format the data or fix the errors found. Make it, it'll be important, it will be really good. So in theory, like, journals or funding agencies require that you make your data public, but it's not really enforced, or at least it's not as useful as it is for us for correctly formatted. So major journals could afford a curator checking standard formatted files publicly available, but that may not happen. So at least we discussed they could have like a checklist for reviewers to make sure that the data is somewhere that is going to be made public in the right format.
There was a question about whether there are downstream databases from EVA that would extract genetic variation from EVA, and we are one of them, like Gramine and some plants. And there is also the other way around, like EVA, it's interested in collaborating with other like resources, community databases to get that information from them. There is, we discussed about these resources, which are really tools. They're not like databases to store VCFs, but it's tools to extract VCF from those, what I understand, but it was just brought up. A killer app solution would be like some place to upload the data files to a DB or to the database, or something like that. A question that if there's a way to use NPL, I think not every, I think I had to escape to be able to see the rest. Let's see. It's not gonna, no. Anyway, I'll just go to the recommended actions. Some I mentioned, make the VCF validator accessible at community databases, or, you know, make it more public knowledge that there is already a validator that you can just, if you formatted your data, just run it through, and it's easy, it makes it easier to submit your variation to EVA. Coordinate with biosamples would be key, because if they make germplasm IDs a requirement, then permanent repositories like EVA, ENA would where we get our data from. Then I think that's the root of the problem there, the basic. There is more education needed to stakeholders about why correct VCF is important and how adding more SNP data can help research and add more value to the data. The importance of having RID markers, and when you're remapping, you know, sometimes, yes, just having an RSID to, even if you lift your data, then that carries along. Or you can have multiple submissions for that same position. You know, it's way too rapid. You have more data for that population. So making those as an educational resource is very important. And for that, we could collaborate with, for example, the FAIR Scientific Literature Working Group you want to hear more about, on ways to encourage authors in journals. We could also collaborate with the Education Working Group, or have look for partnerships with industry, or other experienced educational resources like Ensemble, who have like all these training tools and nice documentation for it. And also we talk about like reusing existing tools from other communities, like the animal and human communities. Okay, so I didn't get to my other questions. I didn't get to organize them, but I think what I mentioned is pretty much answers the other questions: how to incentivize submission of sequence assembly data to the EVA or PubMed, GenBank, strategies for encouraging better metadata submission. So that's, we already covered that, sorry. And that's our group. We have 14 people in person, 5 virtual, and I also asked for the feedback they got there. So try to combine them.
Yeah, let's do sustainability next. Shabari, do you want to come out and report out on that?
I'll do this.
Again, the reason I bought my computer was because we had an additional question that I don't know if it's up there yet, but anyways, yeah, we had 2 groups presenting it. So the first question that we wanted to ask, that we wanted answered was, how can we move forward to a sustainable use and funding for AgBioDatabases? And one of the ideas that was brought up was this question that why are we, instead of scientific research funding infrastructure, can we fund infrastructure to sustain scientific research? Because often the question is, we talk about scientific research, we talk about the importance of that research, and then we say, can we get the funding to build the infrastructure to enable the research? But if the infrastructure is already in place, and we talked about infrastructure setups, like for example, Cybers, which could essentially act as play the role of a hub where we can host multiple databases within them. We talked about investing in workflows that capture how scientists use the bulk download data. We talked about federal funding or growers associations. So we have had some experience talking to various growers associations for various crops. They're very interested in the sustainability idea and want to help and work with the resource to find out how they can enable that. There's also the importance for communicating the research aspect of the database to funding agencies and to funders, because often funders understand that, okay, you're a database. They don't really understand the value necessarily of the research that the database enables. And so that communication becomes important. And so in that aspect, there's overlap with the recruiting and educational working groups as well. And we also talked about the importance of having maybe Hafner carpentry, like data carpentry, things, setups like that, where we communicate to folks how. So for example, if you take a resource like TARE, graduate students know what TARE is. If you're doing research in Arboreopsis or plant genomes, you know what TARE is. How many high school students know what TARE is? Or for example, even CottonGen. Everybody knows about cotton, but how many people know about CottonGen, the database? So let's make tools available for teachers out there who can inform their students about these resources and why they're important. Because people don't think about the importance of databases till they actually have to do the research that informs the database. So let's get ahead of that. The other ideas that was brought up action item wise was building larger, more integrated databases, enabling technical solutions to make it easy to share data across resources. So NRSP 10 was dismissed as an example where there are several different resources that share a very similar platform. And so even though there are very different organisms, if you go to each database, you know how the data is organized in each database. It's organized in a nice, clean way. And that allows also for cost sharing because these are shared resources. That's infrastructure. Standardization, the concern that was brought up was standardization makes things easier, but there's a concern that we might lose some creativity in that. Also, the additional aspect is if you have a shared infrastructure between, let's say, all the AgBioData resources and funding to come for that, how does funding then go? How does it get distributed among the groups? If there's shared infrastructure, yes, fine, but what about funding for other aspects? And also the question was about, we also, keeping in mind about the importance of keeping data, about the resources fair as possible. And also we talk about sustaining the current databases that we have. We also have to keep in mind about new resources that are coming, new databases that are coming, which might not have to worry about sustainability now, but they certainly will have to worry about sustainability in the future. So it's good to have that something in place that will address that issue. The other question we discussed was, can you recruit more scientists and educators to use databases in their work? The key aspect is, of course, to communicate to users, understand the importance and put a dollar value to that, to understand the value that these resources represent to the users. Also about protecting data. One of the key aspects we talked about was about the national security aspect, because there are crops involved in here. And so often we find that these freely available databases, anytime they do an update, within half an hour, all that data is swerved. by databases outside of the US, which is fine as long as they pay for it. So we have to find some kind of system to make that happen. And that conversation can also be had, needs to be had at the level of the government, but we have that conversation about being a national security issue, not just funding research. And one additional question, which, sorry, yeah, just clicking the wrong. One additional question that we discussed was commercial funding, which is looking at commercial, is there a commercial solution that we can look at that would maybe not fund all of it, but at least provide some, in some part, funding for AgBioData. And the first thing would be to communicate with PIs and understand their willingness to do this, because most people believe in fair data and free data, but I believe in fair, maybe not free, but so it's important to understand the value of keeping the resource there. So, and that requires engaging with commercial sources and understanding what are they looking for. Curated data sets, for example, specialized curated data sets, and how can we develop that? How can the resource develop that for the commercial entity, but also do it within the principles of fair data? For example, maybe have a moratorium. Like this data will be made free, but a year after or something. So these are just a quick overview of some of the things that we discussed.
Thank you. Next up, we are going to move on to fair scientific literature with Jacqueline.
It's this one.
Go to the next slide. Please. No.
Do you want to?
Maybe it's a different one.
Yes.
No, I thought I was using the right one, but maybe I just. I'll send it to you on Slack.
Yeah, don't worry.
No, I wasn't asking about.
Okay. Don't worry, nothing happened.
We have all technology.
Yeah, nothing happened.
Awesome. You get to listen to me. All right, so we did actually have a very active group, and I did check some of the comments online also. So thank you everybody online. I really appreciate your comments, and thank you and everybody in person. I really appreciate your comments and your ideas, and we had a great session of basically dreaming big and thinking outside the box. So that out of the way. First question, basically, if there was a tool that could help authors, publishers, librarians, and any of our stakeholders to determine where data should go, what should it look like? Also, because well, there technically is already some of these things. However, none of them really truly do are the dream tool that we're looking for. And so we're like, okay, what else could we have? Talking about that, but then also why not have more creators, biological curators like myself or others actually in this full pipeline of publication to get people in the beginning, even in the beginning when they're starting the paper, talking about organizing the data in different ways. So for different, for ideas, I thought maybe getting a guided submission format where you figure out, okay, you figure out what type of data is, what format is in your data, and then have a validator for your data. What species does it connect to? And then have a help desk, have basically in one site, a dream site that can basically go like a decision tree. And the tool should be really easy and very helpful, almost like the field book. So, and it should be user specified, what do you want to put in and churns out metadata that you were really collecting and shares that metadata. To go further with that, question 2, thinking about the barriers and incentives of making published data fair, what are we missing? So we started with start at the very beginning when the data is even being created. How do we get it from the lab notebook to really get from the very beginning? So there's a whole bunch of different programs. There's e-notebooks, there's protocols.io, there's a whole bunch of different things. But how do we really easily get that from the scientists? And then how do we make incentives? You know, maybe we need to like start making reports to the funders to know, hey, this data was published or this data wasn't published or what it made public. How do we tell the community databases that this data has been made available? Or how do we tell journals or different databases or journals or authors, hey, this data is now available? And then basically like the importance of training throughout, like trying to teach a new tool, trying to teach them about metadata, trying to teach various things, because everybody's time is very limited. So some of the ideas we had was, you know, develop something that would make their research better and easier. Ask researchers really about their workflow and their bottlenecks. And mentioned actually there's a lot of studies in human-computer interaction that we can look into to see how can we really make something worthwhile. There was a great idea about basically making a competitive seed grant money for graduate students to kind of almost develop their own tool that they need to successfully do their research. This does require some planning. Then what are the incentives for publishers? That's the question mark there. Increasing their impact factor, because maybe that increases their citation counts. Like the publishers seem to be the hardest one to really figure out an incentive for. I mean, some others we were able, but. And then what is the most important challenges to address? We also changed this a little bit to what is the most imperative, as in like now, that we need to address. A couple of these comments are from online. I like this one from Peter. The scientific community only cares about their own research and their next grant. Funders and publishers need to stop people from letting researchers get it so that they just don't care about the fair, because all they care about is like furthering their career. Need more incentives to provide, not only just provide data for databases, but the metadata. And then, we cannot throw this all to the reviewers. They do not have enough time to do this. Again, it's the metadata, it's the time. And then making sure data is really available, like accountability at every level. Like we need to be able to do this. So we did think of some low-hanging fruit. That was a good start. Maybe some copy editing. Scan a manuscript to just check. I think I spelled that wrong. Looking at that, like what? Check for like the bio, like probably to see if like what is available. Make sure the data is there, the links work, and then really like automate checks. I mean, with the advance of AI, I mean, can ChatGPT do this for us? They can write code for me pretty well. And I still need to check it. So these are what we had for our discussion. And again, thank you for your time.
And we make it very short because you guys we want to eat. It's lunchtime. And oh, do you want to do it, John? You sure you want to do it? Okay, so I did a report. So I didn't edit so much as you guys have done, and wow, I'm so impressed. But we had a very great discussion in the breakout room, and I also want to thank Rita that she was the host of the online breakout room. So thank you for taking notes. Also, I heard that James helped, and we will look at those during our next meeting. So the first question that we had was like, the problem that we encountered approaching underrepresented communities and what we were doing wrong. And so one great suggestion that one great topic of discussion was like, maybe we are doing wrong because we should build relationship with these communities. And so maybe get inside them, attend their events and go work inside, not from the outside. So I forgot to add it here. So one suggestion was, for example, we can, as AgBioData, attend the next SACNAS event. But please, if you have any other suggestion of this type of workshop or events that underrepresented the institution they are organizing, please reach out to us. Another suggestion was that USDA has established federal liaisons to these communities. So this came from Susan, and so maybe we can invite the underrepresented communities to join our meetings and not to invite people from USDA and hear from them what are their insights about that. And also another topic of discussion is that, okay, we have always the presumption that we have to teach these underrepresented communities about what we know and what we have, but we ignore what they are already working on. And so maybe they have some databases that we should involve in our communities, or we should know what they are working on, as like the indigenous people have done with the care principle. And so what we want to do, what we should do is like reaching out to them to see if they have any and bring them in the discussion about the development of standards. The second question was about how is the best way to serve the needs of our diverse community. And so one problem that we discussed was that in the past, some members of the working group, especially those from very far away countries or underrepresented groups, they started joining the meetings, but then they drifted off because maybe they had some technological issue, really like attending the meetings. We had also the major issue with one working group that the two chairs were from these groups, and there were some problems in that sense. And so we had like so we thought that maybe sometimes the working group, they are task oriented instead of community oriented. So we should make people welcome besides their knowledge and their background. So we don't need to expect people to do their task, but maybe people they just want to join the meeting and learn from what we are doing or what the working group is. And so this will be very important for the next year when we will work on the working group description, and we should not have some requirements about what people will bring to the discussion. Regarding another key point was understanding the needs of the different stakeholders. Before establishing what service AgBioData can provide, we should hear about the needs of the stakeholders. So I have seen before with the Fair Scientific Literature Working Group, we were talking about the publisher, that they are not so many in our community, but maybe we should we should reach out to them and see what are their needs, because probably if we can cover these needs, they will be more motivated to join our community. And we should do this with the different stakeholders. And so also what the database need to be more involved and more actively participating in our consortium, or what the early career researcher need to be more involved in our and so I think this is a very good suggestion that the working group will be working in the next, in this year. And finally, we didn't have so much time to talk about this, but this is a question that we have for everybody here. So if you know about some ag data management groups that we should approach and contact, please let us know. So first of all, I think related with the first question, we should approach databases for underrepresented communities that we ignore, and then they can bring very important insights. And we should engage more with the animal scientists. So one suggestion that actually John had was to put budget in future grant for booth representation in specific event. And he suggested to attend the animal and dairy meeting where we can talk about. And also John will be presenting in July about AgBioData in one of this meeting. And so we are thank you to him. But also another question that was raised from Monica was like, are we still focusing only on GGB databases? Do we want really to broaden our consortium and how much we want to do that? And so this is a question that I hope will make us to think more about it. And with this, I finish. So you guys